Hello, everyone, and welcome to our webinar for 2025. This is a Neurosom and BEL webinar. And today's webinar is Open Science in Sleep Research, from open data sets to accessible pipelines and insights. Today's featured speaker is Rotem Falach, neuroscientist and PhD candidate in Professor Yuval Ner's lab. And this webinar will showcase how HD EEG analytics are advancing our understanding of sleep-dependent memory consolidation and Alzheimer's related neurophysiology. The session will feature applications of SleepEEGpy for sleep EEG preprocessing and event detection, findings from sleep memory and intracranial EEG studies, and discussion from the NIRS lab, Alzheimer's research, and future directions linking sleep biomarkers with cognitive health outcomes. Neurosom's vision, transforming scientific insight into scalable precision neuromedicine. HD-EEG, delivering precision diagnostic biomarkers. And SleepWisp is personalized EEG-guided, AI-driven neuromodulation therapy. And now I'll turn it over to our speaker, Rotem Falach.
Hi everyone, thanks a lot for inviting me for this webinar. My name is Rotem Falach and I'm a PhD student at Yuval Near Lab. Today I'm going to share a few projects that are close to my heart from our lab, mainly about open code and open data, which means open science in general, and how it can make sleep research simpler and more reproducible. So let's go ahead. So a bit about me before we dive in. My background is I did my bachelor's in computer science and I have 9 years of experience as a software engineer, also as an instructor, and for some episodes also a team leader. At some point, I tried to find more interesting stuff to deal with, and I started a master's degree in neuroscience at the lab. That's actually when I started. Almost like 5 years ago, 6 almost. Currently, I'm a fourth-year PhD student, and my research domains are mainly on sleep, epilepsy, cognition, and lately also hyperarousal. I'm also teaching a bit Python and also a TA in the clinical neuroscience track at our faculty. So for those of you who are not so sure why sleep even matters, I guess everyone are in this loop, but just one brief slide about that. So long ago, sleep was thought of like a passive state, like the brain is shutting off kind of thing. But now we know it's the opposite. Many things happen during sleep. For example, some vital routines like synaptic homeostasis, when we reset the connections between our neurons, and also some metabolic clearance, like taking out the trash from the cells outside the body. And also we know the sleep is very much connected to the next day function. When these routines that are supposed to happen during sleep runs well, then our next day is much better. For example, the memory is more stabilized, the attention is more steady, the emotions are more regulated. We could be very angry or sad when we're tired. And also many executive functions work better. In more clinical aspect, we can see some maybe early biomarkers. So sleep is kind of like a non-invasive biomarker we can use to diagnose some neurodegeneration or psychiatric disorders. For example, REM sleep behavior disorder, RBD. When a patient gets this and he starts to move during his REM sleep. So when you have this diagnosis of RBD, the majority of these patients, after a few years, will also get another diagnosis of Parkinson's disease. And this is just one example of a thing that is changing in sleep even before the symptom starts. So what is the challenge of sleep research and what are we doing here today? I wanted to start with like a short story of me starting my master's. So as I told you, I knew Python pretty well and a lot of other coding languages, but I knew nothing of sleep. I just knew it's very interesting for me. That's why I choose this lab. And I also didn't know any of EEG or signal processing. And every student in the lab have different background, right? It's pretty common in academia, different gaps for each one of us, and everyone go through another path. And we have many options how to deal with these gaps. We can take courses, we can use some tutorials, we can ask our friends, our mentors, PIs. Today, of course, we have AI, which is much easier for everything, but we need to be careful with that. Um, and uh, and that's how we gather some pieces together into our new research skills. So I did it. It took quite of time, and also for sure I'm still learning things that are new findings, new methods. And then after a few years, I started to also mentor students, and I saw both undergrad students and master students have the same, we can call it problem. At the beginning, I didn't saw it as a problem when I went through it, but then when it happened again and again, I saw it was very inefficient, and we wanted to be more standardized inside our lab. So we had a problem. Each and every new student project or masters, even PhDs that came from other domains, which can happen. They'll build their own messy, complicated workflow, uh, and we saw it waste a lot of time. Of course, we learn through the way some maybe other stuff, but it could waste some time also for the mentors and also for the students. So we had this idea and this vision uh, to make like a single unified pipeline, which means that everyone could onboard much more easily with the same standards, which would be flexible to different projects and for sure will be helpful for reproducibility. So this is like many buzzwords that I'm just putting here. So let's start with like the details. So we wanted the main goal was to be a beginner friendly setup. So we wanted to kind of like put in some default parameters that not each and every one should get into the specifics of each and every spindle detection and amplitudes and stuff like that. So we put on our basic steps that every project and every data set in the lab that come from the our high density EEG uh, hardware. Um, so we have these common things, and we just put them on together in one pipeline. And we use Jupyter Notebook, so it would be half guided. We have some instructions inside the Jupyter Notebook, and also if you know it, I guess you do. So when you run a cell, you get the output, and you run another cell, and you get the output, and you can take a look pretty easily if it's already written to look at your data and results and to understand if it looks in the right way. And in the same manner, we wanted also some kind of a dashboard, which means we wanted a clear visualization of the pre-processing quality, which means we wanted to take a look at some standard visualization and to understand if this data set is good enough, if we see common things that we used to see, and I'll let you see in a minute an example of that. So I'm not gonna get too much deep into the architecture but the idea was that we have two steps, like in every research project. We have the pre-processing of the data, and then we have the analysis. The pre-processing includes filtering and resampling and referencing, also annotation of noisy epochs. It could be automatic, and also it could be just manual. For sure, I didn't mention we use only existing tools. We didn't want to invent the wheel and to include more problems and more wasting of time. There are common packages that are very very common in like EEG research, like M&E. And for sleep, we use many functions from YASA. And for spectral analysis, we use PyPrep. So again, we just took some different pieces and put them all together in one pipeline that is more specialized in sleep and mainly also in the kind of research that we're doing in the lab. So after we clean the data, we can do or not do ICA, and we create this dashboard that I'll show you in a minute. And then if we want, we can do some event detection and spectral analysis. So this is an example of a dashboard. Here you can see in panel A, all the red dots is the interpolated channels. You can see some numbers and some details about the preprocessing. For example, the reference was average, the bandpass filters, and so on. Here you can see the spectrogram and on top the hypnogram. So for example, here you can just look at the data and see if it looks right. For example, you can see these very red areas at the bottom. We expect it to be in non-REM, mainly non-REM 3, because this is when the slow oscillations are the most powerful. Also, you can see this line here. The yellowish line is the spindles. You can see here the red alpha during wake, and so on. You can check your hypnogram if the sleep scoring is fine. And this is the PSD, the power spectral density. Each line is a different sleep stage. And again, there are some phenomena we're very used to see in a nice data set. So here, this is before the cleaning, before we threw out the noisy epochs. Here in white, these are the noisy epochs that were cleaned out. And this is the PSD after the cleaning, which is very, very nice. This is, of course, a good example. So you can see here again, this is the peak of the alpha during wake. This is non-REM spindles. Here you can see that the non-REM 3 is the most high in the low slow oscillations. And this would help us verify that this data set is fine. And is ready to be analyzed in the next step of the project. Depends on the recent project, of course. And also in B, you can see topographies, which is another kind of analysis that we do in sleep. And you can see here, of course, that alpha is mainly occipital, and the delta is more frontal, and the spindles are more in the middle, and REM and theta are a bit more distributed, but where we expect to see them. So when we have this, we can compare for each and every patient or subject. We can compare them and look with our eyes if we find with like 1 or 2 minutes all the things that we use to search. We have this standard. We can compare different phenomena with this very simple tool that we just clean the data and we get this. Yeah, so we also have some extra analysis. So this is like the next step after the preprocessing. You can see here an example of event detection. This is an average spindle. This is how it looks in the spectral domain. And these are different topographies for the spindles. This is in terms of amplitude, when the amplitude is the highest in the middle. This is the frequency, and this is the duration. And we also have some next analysis tool is the spectral tool, but I'm not going to get into it. And at the end, we wanted to check the consistency. We wanted to make sure that our pipeline produce what established tools already produce, for example, EEGlab in MATLAB. So we ran the same recording through both of them, and we saw that this is very much alike. So they line up. And this was for us like a final validation, and we published the paper with all the details. You can find there the code. You can find a few data examples, a nap, a full night, and also an older adult, healthy controlled full night, if I remember correctly. And this is a nice video I want to show you. Yes, not sure I need this music. But this is just from scratch, how you can open and run SleepEEGpy and get a dashboard. So I took the code from GitHub. I created a new virtual environment in my PyCharm. Then I installed the package. Of course, this is a double speed, but again, it's pretty quick. If you don't like PyCharm, you can use, of course, Visual Studio Code, or you can use the browser for Jupyter Notebooks. So here, this is in the browser. I just run everything. We have a few options if you wanna create like automatic hypnogram, or you already have one, if you wanna clean automatically bad epochs or not. Of course, there are pros and cons to both when you do it automatically, so it's less accurate, and it also takes more time for the computer. But if you have like, I don't know, like a student or a technician that can do it manually, could be it could be more precise, maybe. So why open source? Why did we even do it? Actually, the truth was that, as I started to tell you, we did it for ourselves, for our lab. We just understood that it takes a lot of time to teach each and every one from scratch how to start working with the EEG. And when you already have half pipeline, and of course, it doesn't get into the specific research questions, but it gets us to standardization and easier onboarding. And after like 3 projects that were very, very easy to mentor with this tool, we decided that maybe other labs will be interested. And that was the point when we decided to write it up and to publish. And I think the upside is pretty clear. With open code, it's easier to reproduce results. We can compare our methods, and we can also have community. We can have collaboration with each other. We can fix each other bugs, or if someone create a new feature, you can push it to the GitHub, and then other people will use it. And I think the idea is very, very cool. But on the other hand, there are some disadvantages. They're mainly about time, I think. This is a nice meme I found on Google when you search for open source. So of course, open source tools, some are paid. For example, M&E have a paid team to support everything, but we're just a lab that decided to share their tool, and we're trying to, of course, support it, but it's nobody's job. And that's why we need a community. That's why we need each other to make these things broader and stable and more just more good for everyone to use. And here are some applications that we use this library to find. So, for example, one of the earliest projects we did with this tool was this was published by Flavio Schmidig, a colleague from my lab, last year. What I did was to run naps. Healthy young adults came to nap in the lab, and they did some memory tests before and after the sleep. The main thing here was more about the task, because the common tasks in these days, these are just like memorizing word pairs. And here we presented some pictures of animals and some celebrities. And never mind, it's not like the main topic here, but here you can see the same topography that we had before. Right here of the spindle power, it's in the middle, and as we expect. And here you can see that the spindle density, the events per minute, this is positively correlated with the stability of the memory. So this is, for example, one paper that use it, and another paper work on another domain, which is population of older adults and dementia. So here you can see AD is Alzheimer's disease and MCI is mild cognitive impairment. This is the stage when you deteriorate between your healthy controls and the Alzheimer's. So we found that the slow wave trough, which means the negative peak of the slow waves, when it's more negative, then the cognitive score is higher. When it's less negative, which means it's smaller, because when it goes more negative, it means the amplitude is higher. So when it's less negative, this is correlated with lower MoCA scores. This was published by Omer Sharon, also another colleague from my lab, a few months ago. So up to now, we focused on open code mainly and accessibility of this code. So in another project, we took another step, the next step, and shared also the data. So that's also, of course, have many other advantages. So for example, it can accelerate science in terms of we can reanalysis, we can run reanalysis of the same data and check different methods. We can use these common formats to compare results. We can use it to educate. We can take it for training learners and students, and also for establishing machine learning models, because today it gets bigger and bigger and more accurate. And we're going, I guess in the next few years, we're going to use it more and more. And also we can build trust in science in general, the transparency in our methods. I think it's very, very important. But of course, we have to know that sharing should be responsible. We need to have consent of the patients. We need to do the identification. It gets a bit more complex when you try to do that in terms of ethics. But of course, if you do it, it gets a lot of advantages. So what we did, we had these are very special patients. So we have 25 drug-resistant epilepsy patients. They're going through a surgery because they don't respond to medications well enough. So they have these intracranial electrodes. These are all the electrodes of all the subjects. Of course, this is not one patient, but most of them have electrodes in the medial temporal lobe when you see the darker areas. And each contact have, let's say, 5 to 10 electrodes along the shaft from different brain areas, and they sleep in the hospital. This was a collaboration with the UCLA. So half of the patients were from UCLA and the other half from Tel Aviv Sourasky Medical Center. So they slept in the hospital. We collected this data, and then physicians and doctors sat and just annotate for us manually this interictal activity, which is a pathological activity that happens mostly during sleep in epilepsy, and it's debated whether it's also exists in other neurological disorders. But anyway, we then extracted this to bits format, which is the brain imaging data structure. And we have also some metadata with the sleep scoring and some coordinates of the electrodes. I must say it's only a few minutes for each patient, but in terms of the number of events, we have like 700, almost 800 of events. So we used it to train a model, a machine learning model. When he learns how to detect these events, and this is example of some detection. So we can see in green all the true positive, some false positives in pink, and false negative in pink, and false positive in orange. So what can you do with it? What can you do with this data? So again, you can run your own code on this data, or the opposite. You can take the data you can take the code and run it on your data. So it was data together with the code. So you can choose whatever you want to use. We can train and compare different models, and not only models, also different methods. We can use it for teaching, and we can also ask some sleep and epilepsy questions. For example, stage-specific effects, regional specificity, for example, seizure onset zone related to the epilepsy. And we also have a paper which is currently under review, so I cannot share it, but stay tuned. In this domain, we're also going through some extra analysis of this data set. Yes, and for summary, I wanted to sum it up in like in our vision, future vision. So as I said, we can have open data and open code, and it will help science, and it will create more reproducible insights. It will improve, we can improve together the existing open tools. I'm not talking only about SleepEEGpy, it could also be related to M&E and to YASA and to whatever tool that is now exists. I think the community-driven openness is the main thing. We can compare results and we can collaborate together and create standards that will help us to work together with each other in terms of file formats, in terms of, for example, the dashboard, that we can understand together what is necessary. And we believe the openness is the thing that can unite us. And if everyone will share just a little bit, then the field will gain a lot. I wanted to thank the people behind the project. So of course my PI Yuval and the people I shared their papers. So Flavio, Omer, and Maya is my colleague from UCLA, and Gennady was the main developer of SleepEEGpy. And here's my details. Here are my details for more information. And thanks a lot. And I'm open to get questions if you have any.
Great. Thank you so much for this excellent presentation. And now we're going to turn this over to our colleague and computer scientist, Damien Persico, from BEL, who's going to actually present on our our workflow flow, and share a little bit about that program.
Okay,
Yeah.
so yeah, in this quick demo, I will show you how to create a custom workflow in Flow, which is a database and workflow manager software developed by BEL. So Flow enables users to automatically convert Jupyter notebooks into a containerized workflow that can be run via a web interface. So usually, studies are created within Flow, within this interface, to store and manage files associated with participants. And these files, so we can run different workflows on these participants like this. But let's see how these workflows or custom workflows can be built. Let's take a look first at the notebook we are going to use in this demo, using this notebooks feature in Flow. Let me open this notebook. So in this view, we can open different Jupyter notebooks, and for this demo, we can install different dependencies we can use. For example, SleepEEGpy in this demo, but we can install any other dependencies here. And then here is the code of this script, where we can define inputs, outputs, and different parameters that will be used in this custom workflow. So in order to do this, we use different type annotations to define inputs, whether it is input files or input directory. In this case, is a directory which contains the EEG files to be used like this. And then for the outputs, we can define different outputs. In this case, an output directory that will contain the output files, but we can also specify an output file like an output MFF, and we can also define a PDF file as an output. And then we can define parameters that will be used in this workflow, and we can set different default values for these parameters like this. So this is to define input, outputs, and parameters, and then here we have in these other cells the actual code that will be run in the custom workflow. So for this demo, I will be using just only the first stage, the cleaning functionality of this SleepEEGpy package. You can see here the code. We just use the cleaning pipe here, and then we use some filters, features, and then we auto-detect bad channels here. And then finally, in this other cell, we create the output files that will be saved for this workflow. So this is just a simple workflow or notebook we are going to use in this demo. And then let me show you how we can create the custom workflow by clicking on this workflow creation menu item here. Then clicking on this new workflow button here, and then we can define just a name. For example, let's call it sleep. We can define a description for this custom workflow, and then we can either select a notebook or upload the notebook from the local directory of my computer. But for now, let's use a notebook we just reviewed. So we can select my sleep analysis and then click on the create button here. So you can see that Flow backend automatically runs this series of steps to prepare the and containerize this workflow, and then we can use it for we can execute it later. So you can see a new entry is added here with a status of building. After a few minutes, this will be done, and this status will be changed to available. But I already built this workflow. Here you can see I have this pre-built workflow here, so we don't need to wait until this finishes. So let's go to the studies view again. So you can see here I uploaded a directory containing an EEG file. So you can see here I can click on this sleep menu item here, and then let's review first this view. So basically, this flow automatically creates an entry form and retrieves the parameters from the notebook and automatically sets the default values we just reviewed in the notebook for these parameters. So this can be changed, but let's use this for now. And then it is also possible to change the default values that are used to create the output files. So here we can change the output directory file name, the output MFF, or the output PDF. But let's use the default values for now. And then if I click on the OK button here, you can see we can run this workflow we just created. It is in the running state. And after a few seconds for this demo, this will be complete. You see, this is complete now. And if we go to the studies view again, you will see all these three outputs were added to the same participant that was used to create this custom workflow. And then we can use these files. For example, we can open this in BEL view, which is an EEG viewer available in Flow. So these files can be used within the studies view. So yeah, that should be the workflow. If you have any questions, let me know.
That was an excellent presentation.
Yeah, very quick. Yeah.
Yeah. And I think we have some questions coming up. I think actually, Viral, you might have a couple of questions we might want to start with, and we've got some in the chat, and I just want to open it up for anyone else to go ahead and enter questions into the chat while we get started here.
Yeah, thanks, Corinne. Rotem, thank you so much for the excellent presentation. The combination of open data sets, open pipelines, and the intracranial validation that you've done in collaboration with UCLA is exactly, I think, what the field needs, really to kind of make sleep biomarkers actionable, right? In terms of both research, like what you're doing, and also then translating that into clinical insights and applications. And so we're really looking forward to collaborating with you on this. I mean, as you saw from Damien's demonstration just now, Flow is built with one of the core design philosophies of reproducibility. I think you kind of touched on that as well. And so, and as I was thinking more about your presentation, I saw that your pipeline particularly is modular in nature, right? And so that's something that I found really interesting. And I was kind of wondering if there have been research groups or clinical research groups that have been using parts of your pipeline, for instance, in other applications, like for instance, in the context of aging or MCI or just neurodegenerative cohorts, right? Because sleep architecture may be different in these populations, and I think using parts of your pipeline could be really valuable for them. So I'd love to hear
Yeah,
your thoughts.
so thank you first for your compliments. Actually, we're working together with Soraski Medical Center, Ichilov. It's the main hospital in Tel Aviv, and we're working together with clinicians, and we have different cohorts and data sets of different patients. It can include MCIs and Alzheimer's, as I showed in Omer paper. And also we have Parkinson disease cohort, which is on his way to be published as well. We have the epilepsy patients. We're using this tool from different small projects. It's everything starts like a small project, and then it takes some clear direction. So we have a few parallel projects that are now running, a few projects that started and didn't came to a real result that also happens a lot in science. So yeah, we're working on clinical aspects as well. And it all gonna be published, I hope, in the next few months. We'll see. Maybe years. I need to be more optimistic maybe, but yeah, we're working on it. We have data sets of clinical populations as well, mainly in terms of biomarkers. But also there are also some projects, like if we look at more future vision of what we want to do with this kind of stuff. So it depends on different results. But let's take, for example, we found, I don't know, let's say some pathological event during the sleep. It could also be like a slow wave, which is not like it should be, or whatever. There are also some closed loop attitudes of trying to stimulate with auditory or with maybe stimulation, other kind of stimulations. So this tool, I think, his main idea was it came from our lab experience, which is more biomarkers and passive research, but now all the field is going more towards to some interventions, and we're also going to that way. But we should also add some new features to make it like on real time and closed loop stuff. But yeah, there are so many different angles you can take it. Right now, it's mainly on biomarkers, as you said.
Yeah, no, great. I mean, this is one of the directions that we're taking at Neurosom, Rotem, is to kind of understand the insights from sleep neurophysiology, and translating that into closed loop EEG guided interventions through electrical neuromodulation. And we're kind of looking at particularly the transition between N2 and N3 as a window of opportunity to enhance deep sleep with our sleep wisp therapy. And we've shown significant extension of deep sleep as a part of our sort of at-home sleep trials that we've been running over the last year or so. So
Interesting.
that's really exciting for us. And sort of touching upon what you'd mentioned, particularly with your intracranial work, have you noticed any sort of relationship between the occurrence of, let's say, interictal discharges with your epilepsy data and um, local spindle or slow wave events? I mean, in terms of,
So
yeah, go ahead.
actually, we're now checking this angle. There was a paper published recently in Nature Neuroscience was related to kind of like pathological spindles, which are coming together with interictal activity in the medial temporal lobe. The spindles are more frontal and more like memory consolidation areas. And we are also maybe planning to check this also in our data, but for now it was more like a discussion in one of our group meeting. What else can we do with this data? And we saw this paper. It was very interesting to look at. It was, of course, this was with mainly with I think rats or mice, I'm not sure, but also a few patients with depth electrodes. So yeah, we of course suspect there are some signatures of pathological activity in these populations, and we're searching for them. That's a very interesting direction to go. Yeah.
Yeah, your other work also wrote them on the paired visual associate memory task was also very interesting. I think it looked like you observed some spindle density tracking memory improvements in that paper, right? And I wonder if some of these metrics could serve as early biomarkers in the context of prodromal or MCI or prodromal Alzheimer's patients.
I would hypothesize that yes, this data set is mainly healthy young adult, but as I said, we also have other data sets, but also for MCIs and Alzheimer's, and it's right now currently under research. Like it's my colleagues are working on very similar things and even more complex memory tasks that we can detect memory without asking the patients, because sometimes in MCI and Alzheimer's, maybe they don't know if they remember, but they do remember stuff, right? Like memory is not 0 or 1, it's more like a spectrum. Maybe you remember just a bit, but you're too shy, so you just say you don't remember, or the other way around. You could be very confident and say, "Yeah, I remember," and it's not always that easy to manipulate, to check, to assess. So there is another paper, I didn't get into it because right now it's unrelated to sleep, but then the next step is together with sleep. But it's another paper of Flavio, the one that was leading this nap paradigm, when he track eye movements together with videos, when after a few seconds in the video something is happening out of nowhere, like surprising events. And when you see the video at the second time after a few hours, then you might remember that something should happen at the left top corner, and you kind of expecting it to happen, so you look at it before it happens. And this metric, this was already published, just the method was already published. If you're interested, it's called MEGA, MEGA. memory encoding gaze something. I don't remember exactly the thing, but it's under a Flavio name. And this is also another measure of memory. We're also trying to improve our sleep analysis, but also other measurements analysis like cognition and physiological measurements that are going together with it. Because if we want to check the behavior, how it affects the behavior and memory and things like that, we should be able to measure it in a better way as well. So this is another angle of how do you even check if let's go even to stimulation. Let's say you develop something that helps. How do you know it helps? So this is another angle you need to understand better and to make more precise measurements.
Excellent. I'll stop asking questions, Corey. I'm sure there are a couple more
Yeah,
from
I
the
think we can
audience who will
discuss it
entertain you.
for the whole day. Yeah.
I'll go ahead and ask a couple that are in the chat. Um, are there unique processing steps for integrating intracranial EEG with SleepEEGpy?
Yeah. So actually, the answer here is no. We don't have currently support for intracranial because the SleepEEGpy is mainly for the high density EEG. It can be used like without the dashboard and all the fancy things, but you can use, for example, the spindle detection on the intracranial data. You can take an EEG channel and run it, but the whole pipeline is not like it was more built for our let's say traditional research, and not for that. I hope that uh, maybe another student will take it as a project or something, but for now it's not in the top priorities, let's say.
That makes sense. Let's see. So here's another question. Current LLMs can write Python and Jupyter notebooks. Are developers able to integrate AI coding within SleepEEGpy?
Yeah, so okay, that's a great question because today also I'm using LLMs for coding every day, and it has some problems as much as I have like the experience with it. It depends on the tool you use. I think that nowadays, even if you pay, for example, ChatGPT or Claude, um, you get them to some level of coding of understanding, but at some point, if it's like a niche, more like a niche thing, then the API might be broken pretty easily. They're gonna invent some function that do not exist. Of course, we have documentation website, and you can look up all our API, but uh, sometimes you tell them here, look at that, and they're trying, but it's not perfect. So you can use it. I can try to recommend. I didn't use it yet, but I heard a lot of recommendations, including documentation and API issues about the cursor, if you know it. So it should be like much more related to coding, and it should have some abilities of just reading the GitHub of some package, and it just supposed to know how to use it better. So I didn't try it by myself, but I have a few colleagues that try it and have like great recommendations. It's just a bit more expensive, and it depends on what exactly you want to do. So I think the bottom line is that you can try, but I think at some level it will fail at some point because it's a bit too niche. But maybe if many people will start to use it and it will get more standards, then it will fail less. For starting point, I think it's good enough. You can give him a Jupyter notebook, tell him what you need, and it can help in that manner.
That makes sense. Let's see. Will EEG SleepEEGpy be available in Flow? This might be one for Damien, actually.
Mhm.
Yeah. So currently, this package, given that it is open source, can be installed just like I just showed in the Jupyter notebook, but we can include it in a future version or release in Flow as pre-installed package, just as like other packages that can that are pre-installed in these custom images.
Can I
Great.
ask a question?
Of course.
For this flow that you showed, like what packages or pipelines do you use it now? Like what was the main target of this tool? Or
Maybe
is
I
So
it
can take that. Yeah.
already still in developing stage?
yeah,
What is
it's a new feature. Usually, researchers use this feature using M&E package, and other open source packages. But yeah, this is a new feature that it is in the research mode only for now.
But so Rotem, to address sort of a higher level, Flow has been around the software for a number of years now in many research and clinical research laboratories around the world. As part of our high density EEG offering, right? So we sell complete EEG system solutions for research labs, high
Right.
density, and we see Flow as a universal platform also, which plays nicely into the at-home sleep therapy that we've
Okay.
developed, particularly the SleepEEGpy product. And so Flow kind of ties all of that together, both from a researcher perspective as well as from a clinician perspective, because we see Flow as a path for where, in the future, clinicians that can get access to patient recordings at home through a dashboard, through a cloud-based architecture. So Flow is designed such that it can be sort of maintained in an on-premise server-based model. So all of your data resides within the university, as well as on a cloud. So we have both of those options available for customers and clinicians so that you can maximize your collaboration and so on. So the other nice thing about Flow is it allows you to kind of create specific user accounts that have specific privileges. So you can
Mhm.
have a researcher within your group that you can provide access to a certain experiment or a study folder, and the permissions to edit a certain workflow or not. And then you have higher level privileges where you have access to more than that and so on. So it's designed in a very flexible manner. And as Damien showcased earlier, the whole goal for us is to also allow flexibility such that, and open architecture such that we can build this custom workflows into a single click workflow to enhance reproducibility, right? Because as you noted, oftentimes it's one or two members within the group that
Yeah.
understand the code, but don't know what's happening behind the scenes. Then
Mhm.
someone else kind of plays around with it, and then you can't replicate the data. And so this was one of the bases for why we created this custom workflow feature within the Flow software that is going to be available now as part of users that are using Flow in the upcoming release. So that release is scheduled to be out later this month for all of the research customers. And it's flexible enough that you could use it with not just our data, but EGI data and lots of other kinds of neurophysiological data. So it's very powerful from that perspective as well.
Interesting. Yeah, sounds very, very cool.
Great. Well, that seems like a really nice wrap up for our presentation today and all of the things that we've talked about. I just want to thank both of our presenters today and for everyone attending. And we will have this webinar, it's recorded and it will be presented online for everyone to review there as well. So thanks very much, everyone.